In the 17th century, while most of the world still believed that life could only be seen with the naked eye, one man quietly opened a door into an entirely invisible universe. He was not a university scholar, not a trained scientist, and not part of the elite intellectual circles shaping early modern science. He was a cloth merchant, and yet, through patience, obsession, and an almost unimaginable attention to detail, Antonie van Leeuwenhoek would become the first human being to witness life at its smallest scale. Before his work, the microscopic world did not truly exist in human understanding. There were no bacteria, no protozoa, no clear concept of cells moving and living beyond sight. Disease, decay, and biological processes were explained through broad theories, often disconnected from observable reality. Even though microscopes had begun to appear, they were limited, inconsistent, and often treated more as curiosities than serious scientific tools. Leeuwenhoek changed that entirely. Antonie van Leeuwenhoek was born in 1632 in Delft, in the Dutch Republic. His life began without any clear path toward scientific fame. He was apprenticed as a draper, working with fabrics, threads, and trade goods. His daily work required attention to texture and quality, and this likely introduced him to the use of simple magnifying lenses to inspect cloth. What began as a practical tool became an obsession. Leeuwenhoek did not simply use lenses. He began making them. Unlike many others of his time who used compound microscopes with multiple lenses, Leeuwenhoek focused on crafting extremely small, high-quality single lenses. These were difficult to produce, requiring precise shaping and polishing, but when done correctly, they could achieve remarkable magnification with surprising clarity. He worked in isolation. He did not belong to scientific institutions in the beginning. He did not publish formal papers in the way other scholars did. Instead, he experimented privately, refining his lenses again and again, building small handheld microscopes that could reveal details no one else had seen. Then he began to look. He examined everything he could find: drops of water, scrapings from surfaces, plant material, insects, his own body. Where others might have seen nothing unusual, Leeuwenhoek saw motion, structure, and complexity. In a drop of water, he saw something astonishing. Tiny moving creatures, far too small to be visible to the naked eye, swimming, spinning, interacting. He called them animalcules, little animals. Today we know them as microorganisms, bacteria, and protozoa. But at the time, there were no words for such life. This was not just a discovery. It was a complete expansion of reality. Leeuwenhoek was the first person to observe and describe bacteria. He was the first to see single-celled organisms moving independently. He revealed that life existed at a scale no one had previously imagined, a hidden world as active and complex as the one visible to human eyes. At first, others were skeptical. His claims seemed extraordinary, almost unbelievable. How could there be entire living creatures invisible to everyone else? How could a man outside the academic world be the one to discover them? But Leeuwenhoek had something powerful: evidence. He wrote detailed letters describing his observations and sent them to the Royal Society in London. At first, they doubted him, but over time, his descriptions became too consistent and too precise to dismiss. Eventually, other observers confirmed what he had seen. The invisible world was real, and Leeuwenhoek had been the first to show it. His observations did not stop with microorganisms. He studied blood, becoming the first to describe red blood cells in detail. He examined muscle fibers, plant tissues, and the structure of insects. He looked at sperm cells, making one of the earliest observations that would later connect to reproduction and heredity. Everywhere he looked, he found structure where others had seen none. What made his work remarkable was not just what he saw, but how he saw it. He had no formal training in biology or chemistry. He relied entirely on observation, repetition, and patience. He trusted his eyes, but he also tested what he saw, repeating experiments, comparing samples, and refining his lenses to remove doubt. His life became a pattern. Look, refine, observe again, over and over. In doing so, he was not just discovering new forms of life. He was changing how humans understood life itself. Until Leeuwenhoek, the world seemed limited to what could be directly seen. After him, it became clear that reality extended far beyond human senses, that entire systems of life could exist unnoticed, shaping everything from health to environment. And all of this began not in a grand laboratory, but in the quiet, persistent work of a man who refused to stop looking closer. As Antonie van Leeuwenhoek continued his observations, the scale of what he had uncovered began to expand far beyond a single discovery. What started as curiosity about lenses and magnification turned into a systematic exploration of an entirely hidden dimension of life. He was no longer just observing unusual details. He was mapping a world that no human had ever documented before. His methods were simple, but his discipline was extraordinary. Leeuwenhoek worked alone, often in silence, without the structure of a university or the guidance of established scientific traditions. He built his own microscopes, each one carefully crafted, each one slightly different. These instruments were small, sometimes no larger than a coin, yet capable of magnifying objects to a degree that astonished those who later attempted to replicate his work. He guarded his techniques closely. Unlike many scientists who openly shared their methods, Leeuwenhoek rarely explained exactly how he made his lenses. This secrecy added to the mystery surrounding his discoveries. Some doubted whether his instruments could truly achieve what he claimed. Others suspected exaggeration. But the clarity of his observations and the consistency of his reports gradually overcame skepticism. He documented everything in detail. Instead of formal scientific papers, he wrote letters, long, descriptive accounts of what he saw, how he prepared his samples, and how the organisms behaved. These letters were sent to the Royal Society in London, which became the main channel through which his work reached the broader scientific world. At first, the response was cautious. The idea that invisible creatures lived in water, in the human body, and in everyday environments challenged existing beliefs. Some members of the Royal Society questioned whether Leeuwenhoek was misinterpreting what he saw. Others wondered if his lenses were producing illusions. To resolve this, independent observers were asked to verify his claims. When they began to see similar phenomena under magnification, the doubt began to fade. The microscopic world was not a trick of light or imagination. It was real, structured, and alive. Leeuwenhoek's credibility grew, and he was eventually recognized as one of the most important observers of his time. As his confidence increased, so did the range of his investigations. He examined rainwater, lake water, and water from canals, discovering that different environments contained different types of microorganisms. Some were large and slow-moving, others small and rapid. Some rotated, others glided, others seemed to pulse. He was not just discovering life. He was observing diversity at a scale previously unknown. He also turned his attention inward. Leeuwenhoek examined samples from the human body, including blood, saliva, and dental plaque. In one of his most striking observations, he described the presence of countless tiny organisms living in material scraped from his own teeth. He noted how they moved, how densely they were packed, and how their behavior changed over time. This was a profound realization. The human body was not a closed system. It was inhabited by microscopic life, invisible yet active. Although the connection between microorganisms and disease would not be fully understood until much later, Leeuwenhoek had taken the first step by revealing their existence. His study of blood provided another major insight. He described red blood cells as small, round structures flowing through vessels, giving one of the earliest accurate accounts of their form. This helped shift understanding of the body from abstract theories toward observable structures. Blood was no longer a mysterious fluid. It had components that could be seen and studied. He also made detailed observations of reproduction. Leeuwenhoek examined sperm cells and described their movement, noting that they were active and structured. This observation was controversial at the time, as it challenged existing ideas about reproduction and the origin of life. Some scientists resisted these findings, while others began to explore their implications. What is remarkable is that Leeuwenhoek often worked without theoretical frameworks. He did not begin with a theory and then look for evidence. He began with observation and allowed patterns to emerge. This made his work both powerful and limited. He could describe what he saw with incredible accuracy, but he did not always connect these observations into larger explanatory systems. Even so, his influence continued to grow. The Royal Society recognized his contributions, and he was eventually elected as a fellow, an extraordinary achievement for someone without formal academic training. His letters were translated, shared, and studied across Europe, spreading knowledge of the microscopic world. Despite this recognition, he remained in Delft. He did not move to major scientific centers or seek a public intellectual role. He continued his work quietly, balancing his duties as a merchant and local official with his scientific investigations. This independence allowed him to follow his curiosity without external pressure, but it also kept him somewhat separate from the collaborative networks shaping science at the time. His persistence was unmatched. For decades, he continued making observations, refining his lenses, and documenting what he saw. He did not treat his discoveries as finished. Each observation led to another question, another sample, another experiment. His work was not a single breakthrough, but a continuous unfolding. By the end of this period, the impact of his work was undeniable. The existence of microscopic life had been established. The human body, natural environments, and biological processes were now understood to include components far too small to be seen without magnification. The boundaries of life had expanded, and science could no longer ignore what lay beyond the limits of the eye. Antonie van Leeuwenhoek had done something extraordinary. He had revealed a hidden layer of reality, not through theory, but through relentless observation. And as his work continued, that hidden world would begin to reshape biology, medicine, and the understanding of life itself. As Antonie van Leeuwenhoek moved deeper into the later decades of his work, his role in science became something unusual and difficult to categorize. He was no longer simply a curious observer making surprising discoveries. He had become the primary window into an entire unseen dimension of life, a man whose eyes, aided by his handmade lenses, were effectively extending the limits of human perception. What made this phase of his life remarkable was not just what he discovered, but how consistently he continued discovering. For most scientists, a single breakthrough defines a career. For Leeuwenhoek, discovery became routine. Year after year, he sent letters describing new observations, new organisms, new structures, each one adding detail to a world that had not even been acknowledged before his work began. The microscopic realm was no longer a mystery in principle. It was becoming a field of study and practice. He began to notice patterns. Different types of water contained different forms of life. Stagnant water often held dense populations of microorganisms, while cleaner sources showed fewer. Over time, he observed how these tiny creatures multiplied, how their numbers changed, and how they seemed to respond to their environments. Without using the language of modern biology, he was beginning to see ecology at a microscopic level. This was a subtle but important shift. He was no longer just identifying the existence of microscopic life. He was observing its behavior, its variation, and its dependence on conditions. Life at this scale was not random. It followed patterns, even if the underlying rules were not yet understood. Leeuwenhoek also became increasingly precise in his measurements. He attempted to estimate the size of the organisms he observed, comparing them to grains of sand or other known references. Although his measurements were not always exact by modern standards, they represented an early effort to quantify the microscopic world. This was critical because it moved his work beyond description to analysis. He wanted others to understand not just that these organisms existed, but how small they truly were. The scale itself was part of the discovery. At the same time, he continued to investigate the human body in detail. His observations of blood became more refined, confirming the structure and flow of red blood cells. He examined capillaries, watching how blood moved through the smallest vessels. This helped connect the visible structure of the body to the invisible processes occurring within it, reinforcing the idea that biology could be studied through direct observation rather than abstract theory. His work on microorganisms within the human mouth remained one of his most striking contributions. He described layers of tiny living creatures interacting in ways that suggested complexity rather than simplicity. Some moved rapidly, others slowly. Some seemed to cluster, others remained separate. He noted differences in their shapes, their motion, and their distribution, effectively describing microbial diversity long before the concept itself was formalized. This observation carried implications that would only be understood much later. If microorganisms lived within the human body, then they could potentially influence health and disease. Leeuwenhoek himself did not develop a theory of infection or germs, but he had uncovered the raw evidence that would eventually lead to such ideas. He also explored reproduction further. His observations of sperm cells led him to consider their role in the creation of life. This sparked debates within the scientific community about how organisms develop and where life begins. Some theories suggested that entire organisms were pre-formed within these microscopic structures, while others argued for gradual development. Leeuwenhoek contributed observations, but not definitive conclusions. This again reflected his method. He focused on what could be seen, described, and repeated, leaving broader theoretical interpretations to others. His strength was clarity of observation, not the construction of large explanatory systems. Despite his growing recognition, he remained independent in his thinking. He did not fully adopt the frameworks used by other scientists, and he often relied on his own judgment rather than consensus. This independence allowed him to pursue ideas freely, but it also meant that he was sometimes isolated from broader scientific debates. Still, his authority continued to grow. By this stage, the Royal Society no longer doubted him. His letters were treated as reliable sources of new knowledge. Scientists across Europe paid attention to his findings, even if they could not always replicate his level of detail due to the unique quality of his lenses. What made his work especially powerful was its consistency over time. He did not produce a single dramatic claim and then disappear. He spent decades confirming, refining, and expanding his observations. This long period of sustained work gave his discoveries a depth that could not be dismissed. It showed that the microscopic world was not an isolated curiosity, but a stable and essential part of nature. As the years passed, Leeuwenhoek's work began to shift the boundaries of biology. Life was no longer defined by visibility. It was defined by structure, behavior, and interaction, even at scales beyond direct human perception. This idea would eventually reshape medicine, ecology, and the understanding of organisms themselves. Yet, during his lifetime, the full consequences of his discoveries were not yet realized. He had revealed the existence of a hidden world, but the tools, theories, and frameworks needed to fully understand that world were still developing. Others would build on his work, connecting microorganisms to disease, genetics, and biochemical processes. Even so, the foundation was already in place. Antonie van Leeuwenhoek had shown that reality extended far beyond what the human eye could see, and that careful observation could uncover entire systems of life where none had been imagined. He had not just discovered microorganisms. He had expanded the definition of life itself. As Antonie van Leeuwenhoek entered the later years of his long life, something remarkable became clear. His work had not slowed in any meaningful way. While many would have settled into recognition or reduced their efforts after decades of contribution, Leeuwenhoek continued observing, refining, and questioning with the same intensity that had defined his earlier years. The microscopic world he had uncovered was still unfolding, and he remained committed to exploring it as deeply as possible. By this stage, his reputation had spread across Europe. He was no longer an unknown observer sending curious letters. He had become a respected authority, someone whose findings were taken seriously even when they seemed unusual. The Royal Society continued to receive his reports, and his name was now associated with reliability, patience, and unmatched observational skill. Yet he never transformed into a conventional academic figure. He remained in Delft, living a relatively quiet life compared to the public intellectuals of his time. He did not lecture at universities or build a formal school of followers. Instead, he continued his work independently, maintaining control over his methods and the pace of his discoveries. This independence shaped the nature of his contributions. Because he was not bound by institutional expectations, he could follow his curiosity wherever it led. He examined everyday substances that others might ignore: water from canals, scrapings from surfaces, materials from his own body. He treated the ordinary as worthy of investigation, and in doing so, he revealed that the ordinary was filled with hidden complexity. His observations became more refined and comparative. He began to notice how different environments produced different microscopic populations, and how changes in conditions affected the organisms he studied. Although he did not use modern scientific language, he was approaching ideas that would later be recognized as environmental influence and microbial ecology. Life at the smallest scale was not static. It responded to its surroundings. He also continued to improve his instruments. Each microscope he built was carefully adjusted, often designed for a specific type of observation. His ability to produce high-quality lenses remained unmatched, and even as others attempted to replicate his work, few achieved the same clarity. This technical skill remained one of the key reasons his observations were so far ahead of his time. At the same time, his work began to intersect more clearly with broader biological questions. As knowledge of anatomy and physiology expanded, the structures he had observed at the microscopic level gained new meaning. Blood cells, tissue structures, and reproductive elements were no longer isolated curiosities. They were becoming part of a larger understanding of how living systems function. Leeuwenhoek himself did not fully construct that larger system. He remained focused on observation rather than theory, but his work provided the raw material needed for others to build explanations. Without his discoveries, later developments in biology and medicine would have lacked essential evidence. There is also a growing awareness of the importance of scale. Before Leeuwenhoek, life was understood primarily at the level visible to human senses. After his work, it became clear that the smallest components could be just as significant as the largest. This shift changed how scientists approach questions about growth, reproduction, and health. Even without a formal germ theory, the idea that invisible organisms existed within and around the human body could not be ignored. It introduced a new dimension to thinking about disease, even if the connections were not yet fully understood. The possibility that unseen life could influence visible outcomes began to take shape, slowly altering the direction of medical thought. Leeuwenhoek's persistence also created a model of scientific behavior. He demonstrated that discovery did not always require large institutions or formal training. It required attention, patience, and the willingness to question what others overlooked. His life showed that careful observation, repeated over time, could reveal truths that had remained hidden for centuries. This example had a quiet but lasting influence. It encouraged a broader view of who could contribute to science and how knowledge could be produced. The idea that a single individual working outside traditional structures could reshape an entire field challenged assumptions about authority and expertise. As he aged, Leeuwenhoek remained mentally sharp. He continued writing letters, describing new observations and refining earlier ones. His communication with the Royal Society did not stop. Decade after decade, he maintained a steady flow of information, building one of the most extensive records of microscopic observation in early science. His life stretched into his 90s, an extraordinary length for the time. This long lifespan allowed him to observe not just individual samples, but changes over years. He could revisit ideas, confirm patterns, and deepen his understanding through repeated examination. His work gained strength from this continuity, from the accumulation of careful, consistent observation over time. By the end of this period, the invisible world he had revealed was no longer a question. It was an established part of scientific reality. Microscopic life existed. It was diverse, and it was active. It interacted with environments, appeared in the human body, and followed patterns that could be observed and recorded. The boundaries of biology had expanded permanently. Antonie van Leeuwenhoek had not just opened a door. He had kept it open, patiently exploring what lay beyond it for the rest of his life. And as his work reached its final stages, the foundation was firmly set for future generations to move beyond observation into explanation, building entire sciences on the world he had first brought into view. When Antonie van Leeuwenhoek's life finally came to an end in 1723, he left behind something that was both enormous and strangely quiet. There is no single grand theory attached to his name, no unified book that defined his work in one place, no institution built around his legacy. Instead, he left hundreds of letters, thousands of observations, and a completely new dimension of reality that had not existed in human understanding before him. What he discovered did not fade. It grew. At first, the scientific world simply absorbed the fact that microscopic life existed. That alone was revolutionary. But as the 18th and 19th centuries unfolded, the implications of Leeuwenhoek's work began to deepen. Scientists returned to the world he had revealed, this time with improved instruments, new methods, and broader questions. Gradually, connections began to form. The tiny organisms he had described were no longer seen as isolated curiosities. They became part of larger systems. Researchers began to link microorganisms to processes such as fermentation, decay, and eventually disease. What Leeuwenhoek had seen moving in a drop of water or living in the human mouth would later become central to microbiology and medicine. This shift reached a turning point in the 19th century. Scientists like Louis Pasteur and Robert Koch built directly on the existence of microorganisms, demonstrating that they were responsible for infections and could be controlled through sterilization and hygiene. The germ theory of disease, one of the most important developments in medical history, depended on a simple fact that Leeuwenhoek had first revealed, that invisible living organisms exist and interact with the human body. Without that first step, the rest could not follow. His work also shaped biology in a broader sense. The idea that life exists at multiple levels of scale became fundamental. Cells, tissues, organs, and organisms could now be understood as part of a layered structure, with microscopic processes underlying visible ones. The development of cell theory, genetics, and molecular biology all relied, in part, on the expansion of vision that Leeuwenhoek had made possible. Even the tools of science evolved from his approach. Microscopy became a central method across disciplines. Instruments improved, magnification increased, and entire fields emerged from the ability to see what had once been invisible. Today, from electron microscopes to advanced imaging technologies, the principle remains the same. Look closer, and new worlds appear. What makes Leeuwenhoek's legacy especially powerful is how it changes the way reality itself is understood. Before him, what could not be seen was often treated as non-existent or irrelevant. After him, invisibility no longer meant absence. It meant limitation of perception. This shift has influenced not only biology, but science as a whole. It encourages the idea that there is always more beneath the surface, more to discover, more to understand. His life also carries a deeper lesson about knowledge. He was not driven by formal theory or academic expectation. He was driven by curiosity, by the simple act of looking carefully and refusing to ignore what he saw. He did not wait for permission to explore. He built his own tools, asked his own questions, and trusted observation over assumption. This independence made his work both unusual and transformative. He did not follow the scientific structure of his time. In many ways, he helped reshape it. He showed that discovery could begin anywhere, even in the hands of someone outside established institutions, as long as there was discipline, patience, and attention to detail. There is also something quietly profound in the nature of what he revealed. The microscopic world is not distant or abstract. It is everywhere. It exists in water, in air, in soil, and within the human body itself. It shapes health, environment, and life in ways that were once completely hidden. Leeuwenhoek did not discover something far away. He discovered something that had always been present, unnoticed, influencing everything. That realization continues to matter. Modern science constantly returns to this idea, that reality is layered, that understanding requires deeper examination, and that what appears simple often contains complexity beyond immediate perception. This perspective guides research in fields ranging from microbiology to nanotechnology. In the end, Antonie van Leeuwenhoek did not just discover microorganisms. He changed the scale at which humanity understands life. He extended vision beyond natural limits and proved that entire systems of existence could remain hidden until someone chose to look closely enough. His work did not conclude with answers. It opened questions that are still being explored centuries later. Every time a scientist studies bacteria, examines cells, or investigates microscopic processes, they're continuing a journey that began with his lenses. A journey into a world that was always there, waiting to be seen.